In part 1 video on pharmacodynamics, we will discuss the receptor, its types, then we will discuss the two important properties, and then we will discuss the potency and efficacy. In the next video, we will discuss the agonist, antagonist, and many more. So first, let's start this video. Here, this is a cell, and here a molecule is present, and this is drug, and it is coming here, and it is going to bind with this particular biological molecule. As a result of the binding of drug with this molecule, a change occurs in the system. So this molecule is known as receptor. So we can define receptor as a molecule with which when drug binds, a change occurs in a biological system. The biological system can be anything. Here a cell is represented. Suppose this is an SA nodal cell, and the receptor here is beta 1 receptor, and the drug that actually came here is a beta 1 agonist like dobutamine, and the binding of that drug with that particular receptor results in the increase in heart rate. So a change occurs by the binding of drug with a particular molecule. That particular molecule is known as receptor. That's it. So I think receptor is clear. If we zoom it in, then you can see here that this is the receptor and the drug is binding with a specific site. It's not binding here and here. It's actually binding with a specific site, and this site is known as receptor binding site. The receptor can be a protein, it can be a nucleic acid, and it can be an enzyme, but that's not important here. So here this is the inactive state and this is the active state, and four terms are important here. One is agonist, second antagonist, third inverse agonist, and fourth is partial agonist. We will discuss them in detail in the next video, but till then I want to tell you guys that agonist increases the receptor concentration in the active state. Like if I talk about a receptor in the absence of any drug, in the absence of any endogenous particle, 80% receptors are inactive and 20% are in the active state. So when the agonist binds with the receptors, the number of receptors in the active state they increases. Like I have told earlier in the example 80-20, so the receptors in the active state they increases, like they may become 80 and they become 20. So agonist increases the concentration of the receptors in the active state. Next is antagonist. Antagonist actually binds with the receptor and they stabilize them in the inactive state. Inverse agonist. As I told earlier that in the inactive state, in the normal state, in the absence of any drug molecule, we have inactive receptors that are 80 and 20 are active. So the inverse agonist is actually going to convert those 20 into the inactive. So it actually increases the concentration of the receptors in the inactive state. Yeah, that's a little bit different. Yes, next is the partial agonist. So partial agonists also bind with the receptor. It actually converts them in the active state. But the response here is not that much as occurs in the agonist. So that's why it is known as partial agonist. Okay, but we will discuss them in detail in the next video. Till then, let's move further. Here we have four types of receptors: ligand-gated ion channel, G-protein coupled receptor, enzyme-linked receptor, and intracellular receptors. The first three receptors they actually bind with the hydrophilic molecules, whereas the fourth one here it's binds with the drug that are hydrophobic, because as the name indicates intracellular, these receptors are actually present inside the cells. So the drug molecule has to cross the membrane, and we know that the lipid soluble molecules they can cross the membrane easily. Yes, you all knows. Okay. Okay, now here this is our drug molecule and this is a channel, and we will keep this as simple as possible. So this drug come here and it binds with this binding site. After it binds, the pore opens, and depending on the drug and the particular channel, the specific ion can come in or it can move out. Like if this is a GABA molecule that comes here and this is a GABA receptor, then what will happen? The chloride ions they are going to move in, and they can result in the hyperpolarization in the neuron. Or if it is the nicotinic molecule that is coming here and it is binding with the channel, then the sodium ions, they come in, and the potassium ions, they move out. As a result, there will be depolarization and the neuron stimulation. And if this channel is present in the heart, and the particular drug that came there and binds and resulting in the heart contraction, so these are actually examples. Next is channel that is voltage gated. So here in this case, when the drug binds with the voltage gated channel, it actually regulates the channel opening. Like suppose if our channel opens at particular voltage, then after the binding of the drug, it doesn't open at that particular voltage, or the voltage may be increased or the decreased for the opening of that channel. That's it. So the local anesthetics are the example here, and they actually block the opening of sodium voltage gated channels. Next is the G protein coupled receptor, and here this is a receptor. It has 7 transmembrane units, and this is the extracellular domain that is going to bind with the particular drug, and here the intracellular domain is bind with the G protein. G protein has 3 subunits alpha, beta, gamma. On the binding of the drug with the extracellular domain, the alpha unit, it actually replaces the GDP with the GTP, and then it moves towards the adenyl cyclase, and this converts the ATP into cyclic AMP, and this cyclic AMP will cause the conversion of PK, and you all know what actually happens after this. And if this is GQ, then the phospholipid is actually broken down into DAG and IP3, and this DAG is going to cause the conversion of activation of PKC, and the release of calcium channel is caused by this IP3. But the point here that I actually want to tell you guys is that the drug binds with a receptor, and these molecules are produced, and they are actually going to cause a change in the system. So the drug and receptor, they combine and resulting in the formation of complex, and the complex then activates the effector. The effector can be a protein, it can be an enzyme, it can be an ion channel. But a cool point here is that the effector can cause the formation of second messenger, and this second messenger can cause the activation of another effector. Like here in this case, DAG, calcium, this cyclic AMP, they all are second messengers. Next category, we have enzyme-linked receptors. So this is insulin, and it is coming here, and it is binding with this receptor. Here these are tyrosine molecules that represent inside the cell. First, they are not phosphorylated. After the binding of the insulin, they become phosphorylated, and this phosphorylation is auto. So here they are autophosphorylated, and their phosphorylation results in the phosphorylation of many molecules, and as a result, these IRS, they can also become phosphorylated, and when they become phosphorylated, they can actually cause there is activation of a number of metabolic pathways, and as a result of activation of those pathways, there are many metabolic effects that actually occurs. So with the binding of the drug with a receptor, change occurs in the system. Here very important receptors, the intracellular receptor. Okay, so the drug crosses the membrane, enters the cell, and binds with the receptor, and as a result of the binding, it can move to the nucleus, or the receptor here can be in the site, can be present in the cytoplasm, or they can be present in the nucleus as well. But if the receptor and the drug complex, it actually acts on the transcription factors, then there can be the mRNA that can be increased or decreased depending upon the drug and the receptor type, and the DNA of particular genes may be more transcripted, or there is increased expression of, there's decreased expression of some other genes, or similarly this drug molecule can actually binds with this tubulin, and it can bind with the ribosomes that you will study in the chemotherapy. Now, two very important concepts here. One is the tachyphylaxis. So, what is tachyphylaxis? It is actually the desensitization. What is desensitization? Desensitization is that if a particular receptor in the presence of a particular drug or stimulus, it actually produces its response, then if it becomes insensitive to that particular drug, then that is known as tachyphylaxis. By phosphorylation, or it can happen by any other chemical pathway, but the particular receptor, it become insensitive, desensitized to that drug. Next is upregulation and downregulation. So if many drug molecules are present for a long period of time, then the number of the receptors they actually decrease down, and the upregulation is the opposite. Like if number of the drug molecules or the endogenous particles they are low in amount, and the receptors they actually increase in their number, so this is actually up regulation of the receptors. Next is the refractory period, that is also a part of this regulation. Here refractory period is important in case of the ion channels, because we know that after a channel it closes at a specific voltage, then it will take its time and then it will open, and that's very simple and it doesn't need to be discussed in detail. Next, a very beautiful concept here is signal amplification. We have two drug molecules and they are binding with two receptors. As a result of their binding, suppose this is G protein coupled receptor. The G proteins are activated and their number is more than that of the receptor number. Then this G protein they are going to cause an increase in cyclic AMP, and this cyclic AMP is far more than the one that is produced by here and then here. So and these are going to cause a lot more response. The point I want to tell here is that the number of receptors they are small but the response is very large, so actually a signal amplification occurs, and this signal amplification is responsible for the presence of spare receptors. And the spare receptors, why they occur? Because suppose there are 10 receptors and the 10 is a very low number. Suppose there are 100 receptors for a particular drug, and the maximum response because of this mechanism, because of this signal amplification, actually occurs only by the binding of drug with 50 receptors. Then the remaining 50 receptors they are known as spare receptors. So that's important, because here in case of insulin we have 99% of receptors that are spare receptors, so the actual response occurs only by the binding of insulin molecules with the 1% of receptors. But here in case of the heart, the beta receptors, there are only 5 to 10% that are spare receptors. Next, we have potency and efficacy. Whenever the term potency came in your mind, always remember drug concentration, because potency deals with the drug concentration and it is used in the comparison of two drugs. Like there are two drugs and they produce a particular response. Like if I say that there are two drugs, like this is the candesartan and this is irbesartan. 4 to 32 mg of candesartan is required to produce the same response when 75 to 300 mg of the irbesartan is given. So, which drug is more potent? The candesartan, because a small amount of drug is producing the same effect when the irbesartan is given in large dose. So that's the potency. So the drug candesartan is more potent than irbesartan. This is the graph. It is known as dose response curve, and here on x-axis we take the dose and it is increasing in this direction, and on the y-axis we have the percentage of the response that occurs after the binding of the drug with the receptor. So in the initial stages there are far more free receptors, and the drug molecules they came and they bind with the receptors, and there is a sudden steep response. But with time the receptors they are occupied and the less free receptors are present, then the drug binds and resulting in this. But here you can see this is the maximum response, and there are two drugs, and this is this point, like the 50%. This is the Emax, like the maximum response, that is known as Emax. And if I talk about like EC50, the 50% of response, so it can be drawn like this, like it can be here approximately the 50% response. Okay, so this is the drug concentration that produces the 50% response. And if there is another graph here, let me draw. Both the drug actually produces the same maximal response, but here for this drug A, this is the concentration of the drug that is required to produce the 50% response. But in the case of the graph B, we have this amount of the drug that is producing the 50% response. So which drug is more potent, the one B and the one A? Yes, you are right, A. This drug is more potent. Next is we have the concept of efficacy. So what is efficacy? Efficacy is the maximal response, like the Emax, the 100% response that occurs here. Emax. Here the border drugs have the same Emax, but this is not the always case. Like maybe the second drug B doesn't have the same Emax, and both they are same. They have the same potency, but they don't have the same Emax, or they don't have the same Emax but they have the same potency. That can occur as well, but we are not discussing it right here. The efficacy, it depends on two things. One is the drug receptor complex formation, and the other is the intrinsic activity. Like, if we all know that the binding of the drug with the receptor is essential for the response, and the more the complex are formed, the more the response. Okay, that's it. Okay. But another point here is, why if the partial agonist binds and the response is 70% response, and when the full agonist it binds, the response is 100%. In case of both, the receptor drug complexes are formed, and suppose there are 100% formation of the complexes, but why the response is 70% in case of partial agonist and 100% in case of full agonist? So that's the intrinsic activity. Okay, so if we talk about the partial agonist, then the response is less as compared to the full agonist. But in case of antagonist, we all know that it binds and stabilizes the inactive state and doesn't allow the receptor to bind with the drug or with the endogenous substance, and it actually blocks the response that is caused by that particular drug. Next is the inverse agonist, and we all know that it actually increases the concentration of the receptors in the more in the inactive state. It actually converts the active receptors that are present in the absence of the drug in the inactive. Okay, but in the next video we will discuss the energy relations, the receptor binding, and the agonist and antagonist, and many more. So if you like this video, click on the like button and also subscribe to the channel. Thank you so much for watching this video, and click on the bell icon to stay connected. Thank you so much.